And shall I start?
Yes, sir.
And partakers. Good morning. Thank you to everyone who joined us this morning over online meet. It's a glorious moment to extend my warm wishes on behalf of the Sri Ramakrishna College of Arts and Science, Coimbatore, formally secured grade A+. It's recognized by the UGC Act from 2007 onwards to till date. It is an ISO 9015 certified college. The college is under DBT, Government of India. At present, there are more than 5,500 students pursuing education in 33 different UG and PG programs, and 12 PhD programs also going on. We all are yet part of this institution due to our shared vision for the vision of the institution, our passion unites us, and our hard work, which strengthen the institutions. Our department, technology, is serving the academic needs of the institute since 2006. BSc, MSc, MPhil, and PhD full-time and part-time programs. The main motto of the department is to make the students to express their innovative knowledge and scientific skills for the new era of biotech. Auspicious, the one-day international workshop on biological research and innovation industrial revolution. The key impact on industrialization. The program organized by the Department of Biotechnology, Ramakrishna College of Arts and Science, under DBT-SRCAS scheme, which is funded by DBT New Delhi. On that note, it is a great occasion to welcome our guest of speaker today, Ms. Sri Suresh, scientist to Thermo Fisher Scientific, California, US, who is a notable alumni of our department and studied BSc Biotechnology 2014 and 2016. We are proud to spell out our guest, who is a scientist in Thermo Fisher and industry expert. And she will be giving a lecture on COVID research and frontiers in biotech industry. It is a very useful topic for all of us. So we are grateful to her for accepting our invitation and becoming a guest of today's workshop. I feel delighted to welcome you. Then the afternoon session, which is entitled Future of Food, Challenges and Opportunities in Industrial Revolution. It will be given by Dr. Lakshmi Mohan, Assistant Professor, Department of Food Technology, St. Eggs College of Engineering, Kottayam. I want to convey my heartfelt gratitude to our beloved Principal and Secretary, Dr. B.L. Shivakumar, sir, who is keeping on motivating us all enough by being an example of light in the darkness. We would be delighted with his expertise and knowledge in every way possible. Also welcome our Vice Principal, Deans, Directors, HODs, and faculty members of Biotechnology and other departments of Sri Ramakrishna College of Arts and Science, who is participating with all us. I extend my wishes and welcome to today's program organizing secretary, Dr. D. Jayashila, who is arranged this event. With this, I extend my hearty welcome to all the faculty participants and participants from various parts of the country and students of host department and other departments who wish to participate in the program and utilize the knowledge as pleasurable one. Without any further delay, I would like to start the events. We would be delighted to have your suggestions and questions in the way of Thank you.
Thank you, sir. The industrial is further improving the competitiveness of the bio industry, leading to next generation economy and the goal of realizing a globally advanced bioeconomy in 2030. So the resource persons of today's workshop, Dr. Lakshmi Mohan, who will be joining soon in the session 2, and Ms. Sri Suresh is expertise in enhancing and facilitating the R&D for establishing advanced basic technologies. With this brief introduction, I would like to introduce the resource person of the first session today, Ms. Sri Suresh, Scientist to Thermo Fisher Scientific, California, USA. She is a proud alumni of the department. She has completed her Bachelor of Science Biotechnology in School of Science and Humanities, SRCAS College of Arts and Science, the former name of Sri Ramakrishna College of Arts and Science. She has completed a Master of Science in Industrial Biotechnology in School of Biotechnology and Genetic Engineering, Badia University, and Master of Science in Biotechnology, Graduate School of Arts and Science, Georgetown University, Washington, D.C. She is currently working on developing stable cell line for SARS-CoV-2 mRNA vaccine production using TALENT technology, site-specific integration of genes, and stable expression of cell lines. This development would lead to high yield cell lines and high quality product for vaccine development. She is also skilled in maintaining mammalian cell culture and expertise in protein analysis. She is also the Associate Research Scientist in BioQuell INC. She has established a project entitled Viral Fusion Mediated Cell Signaling in SARS-CoV-2 and HGP1 in Cardiovascular Pulmonary Pharmacology Research Laboratory, Georgetown University Medical Center, Washington, D.C., US. She has trained more than 15 undergraduate students to conduct experiments involving plant tissue culture, molecular biology and PCR, HPLC, ELISA, western blotting, protein purification, and flow cytometry during her work as research assistant in ICAR Sugarcane Breeding Institute, Coimbatore, Tamil Nadu. She has presented papers, posters, and publications related to the research on SARS-CoV-2 spike protein and other lung vascular cells. She has also participated in many conferences and workshops related to healthcare research and development. She also holds astonishing contribution in public community, like President of Rotary Club of SRCAS India in 2015-16, Club Advisor of Rotary Club of SRCAS in 2016-17, Chairperson of Organ Donation Rotary Club, SNR Trust, which is a remarkable event of Guinness World Record in 2016, and Brand Ambassador of SNR College, Coimbatore. I feel very happy and delighted to have her in our midst. Ms. Sri Suresh, the session is all yours. Thank you.
Thank you so much. It's good to be back with SNR again. I'm so obsessed with the name SNR, and I'm still not able to get the word SRCAS. I'm just getting used to it. I'm so happy and thankful. It's been 4 or 5 years talking to you all. It's good to be here. So I would be happy if you all can turn on your cameras. I'm not going to put anyone in spot, but then I would love to see all your faces behind the screen I just feel like talking to humans and not to just a computer screen. So I'm just gonna go ahead and start my session real quick. So can you all see my screen?
Yes, ma'am, it's visible.
Okay, that's cool. Okay, so I'll get started. So this is Sri Suresh, and I'm so proud to be an alumni of SRCAS College. And that SRCAS College has been a great platform for me. And I'm happy from moving from SNR to US. And I would always say I'm a proud alumni of SRCAS College. So I'm currently scientist working in Thermo Fisher Scientific, and I currently work on COVID vaccines. So I'm pretty much working on COVID like last 2 years, or probably since all this SARS-CoV-2 thing started. So I was earlier working with live COVID cells in the cell-free environment, like in testing vaccines in animals, testing the vaccines of Pfizer, Moderna, AstraZeneca vaccine, probably earlier this year. So last 2 years, my journey was literally living with COVID, and it's still continuing. I hope this ends soon. So to get started, by just seeing the picture, you all would have figured, what I'm going to talk about. So it's something on SARS-CoV-2. So I think you all would have just gone through or seen this picture earlier. So we're all just talking about SARS-CoV-2 for the past 2 years. What happened to the other viruses and bacteria? Are they just waiting, or are they really surviving, or what's going on with that? Let's get into it. So there are three classes of the viruses that were recently discovered in humans, which are, the re-emerging of these viruses. One is obviously the influenza, and the other one is very obvious, the SARS-CoV-2. So the other re-emerging outbreaks that would seriously cause an epidemic, virus, Ebola virus, and still having cases of Zika, Ebola, or measles, but they are not as big as pandemic or epidemic. But there are still cases going on. It's just submerged because of the COVID thing that's going around. So why is this SARS-CoV thing is not ending? It's been 2 years, and we are so developed in all this biotechnology industry, all this research. We have a great R&D all over the world. Why is it not ending yet? Even in this 21st century, we're still suffering all these diseases, and it has killed more than 4.5 million people around the world. Why? So these viruses usually tend to routinely jump from one host to the other. So say, for example, they tend to move, the SARS-CoV-2 tend to move from like the bats to humans. We just keep switching receptor called as ACE2. So that receptor is responsible for that in the later slides. So why these RNA viruses change real quick, and this makes the viral infections extremely difficult to detect or treat or even prevent them. So the high mutation rate of RNA viruses is increasing the ability of these viruses to adapt in different, as you could see, the one nucleotide per genome is first time it's one, then it replicates into two. So it just incorrect genome, and that's the reason why we are getting different mutated genes. So these are particularly called Coashi species, meaning the individual viruses species of population each have slightly different genomes. It's the same virus, but it has different genome. The genome that are different was incorrectly reproduced. So that's how we get a lot of mutated versions of SARS-CoV, of COVID viruses that we have till now, and it's still being added. The main two things are the severe acute respiratory syndrome, which is the SARS-CoV, and the other one is MERS. You would have probably heard of this. It's the Middle East respiratory. So these two viruses are particularly the most very hurtful viruses for humans, and they are because of epidemic or pandemic. So these are the list of viruses. Specifically, MERS and SARS both were, from bats. They just originated from bats. Indeed, about coronavirus, You could always stop me in between if you have any questions or if you want anything. I don't think I can see your chat, but if you have any questions, feel free to unmute yourself and ask any questions. So, what are these coronaviruses? These are RNA viruses which has real large genomes. So, and we call as a coronaviridae. So, they possess a very large, about 30 kilobase of their strand, and they are single-stranded, obviously, and it's an RNA genome. So these the SARS-CoV is not new now. New now. It's already emerged in 2002, and it just rapidly spread all over the world. As not as big as with 10, it had 1,000 cases in 2002. So the SARS not just in 2002, it was still there earlier, but they were never considered as they were just as common as a cold or a cough in human. Now, after a great mutation, it's now reached to make it as a pandemic. So that's how it is. And let's talk about the protein code. That's very complicated. That makes things complicated for people who are working on vaccines. So if we usually know the virus genome is of either DNA or RNA that's inside it, and then it's coated with a protein. Sorry. Give me a second. Sorry about that. Okay. So we know the genome is either with DNA or RNA inside, and then it's coated with a protein shell outside. So these viruses are completely dependent on replication. If you just stop the replication, they would never grow. But you could never stop replication. So that's the hardest part. So these viruses come in different sizes or shape with respect to the DNA or the RNA genomes, with or without the envelopes. So we all have also seen viruses that do not have any outer membranes, that is without any envelope or without any protein code. So the RNA viruses have a compact genome, and it has very high mutation rate than any other microorganisms. So it's always it's a tricky thing to handle this. So we got to know about the spike protein a bit more, and a suggestion or answer from what you've understood. I'm not going to put everyone on spot, but then just be sure I'm just gonna. So we all know the SARS-CoV-2 is causing the current pandemic. It has affected more than 219 million people. It was actually 218 million people when you know, when I was typing this, and I realized it came to 219 million in just one day. So, and it has killed more than 4.55 million people, patients, or so far. So, what's the key function coronavirus? As I said, it's this spike protein. It's this red things that's creating a lot of troubles. Do to your cell. So many scientists are still trying to understand the infection mechanisms of SARS-CoV-2 and viral protein, which is the S protein. That's the outer membrane, or it's called the envelope. It's a class 1 viral membrane fusion protein. So the process by which the enveloped viruses enter the host cell is called the viral membrane fusion. This is the host, and this is the SARS-CoV-2 spike protein, and this process is called as the viral membrane fusion, by which the fusion of the virus and the cell membranes result in the release of the virus into the genome of the cell, and that's how it works. So this characteristic crown-like appearance that you see here is called as the corona. Facilitates the virus to enter into the host cell, such as the endothelial cells or smooth muscle cells, etc. So the adverse effects of COVID-19 are particularly potent for elderly people, and also with people who have pre-existing cardiovascular conditions. So as you could see, it has affected more elderly people than younger people. And then it also has affected people with heart diseases and lung diseases. So why did that happen? So recently, it was reported that the SARS-CoV-2 spike protein itself, that is, um, and again, I said that. I'm sorry. Okay, so the spike protein itself, without the rest of the viral components, is enough to elicit cell signaling in lung vascular cells. And you don't need the whole viral genome. Just the spike protein is enough to cause you positive for COVID-19, or can cause any type of viral infections, or particularly lung infections or lung in the lung vascular cells. So why is this so, you know, so good in doing this? So in this study, it was said that the treatment of the human pulmonary artery smooth cells, that's in the figure A, and then or the pulmonary artery endothelial cells in the figure B, with the recombinant SARS-CoV-2 spike protein at the very low concentration, it activates the MEC, which means it without the rest of the viral components, just that the spike protein itself can elicit cell signaling, and that could cause severe diseases in humans. Why is this happening? So when the activation of cell growth signaling in the lung vascular cells starts, the SARS-CoV-2 spike protein especially hurts the pulmonary vascular walls in humans. So patients who died of COVID-19 are seen to have a thickened in the pulmonary vascular walls. So why did this happen? So in this study, we just researched a bit more and found that since the SARS-CoV-2 spike protein can elicit cell signaling, that can cause severe effects on vascular walls, and that's the reason why patients who die of COVID-19 have thickened pulmonary vascular walls. So it's not the viral component; it's only the spike protein that's causing those lung vascular diseases. But however, H1N1 is kind of similar to what we're talking about COVID-19. They also have a spike protein, but they do not elicit any cell signaling in human vascular cells. That is, they don't elicit any cell growth, or they don't trigger anything in the human vascular cells, whereas SARS-CoV-2 does that. And that's why it makes it even more complicated. So we created a viral protein fragment theory to know what exactly is happening with SARS-CoV spike protein. So based on these results, as I said, we proposed a viral protein fragment theory, which states that the cell signaling elicited by a protein, that is a spike protein, that occur in cardiovascular cells would predispose or infect individuals to develop complications that are seen in several, or severe fatal COVID-19 conditions. So that's a boy, that's a guy that we need to target in the future. From the before mentioned theories, we know the cell signaling elicited by the spike protein and its fragments, the cardiovascular cells, also can predispose severe diseases in individuals. So thus it's very important to know what is the spike protein's role in the viral membrane fusion, and what does it do to elicit cell signaling, and how does this affect the rest of the host cells? How is this working? So what we try to do is this only the SARS-CoV-2 that's causing all this? What about the rest of the viruses? What is happening with HIV? What is happening with H1N1? What is happening with Ebola? What's happening with Zika? What's happening with dengue? So we just try to figure out what's happening, or it's just only a specialized character in SARS-CoV-2. So what we did is we did several PubMed and Google searches we performed. We read thousands of papers, try to analyze the experimental results with a viral membrane fusion of rest of the viruses other than SARS-CoV-2 or SARS family or the COVID family, elicit cell signaling that could cause severe diseases in lung vascular cells. But unfortunately, as you can all see, except the SARS-CoV-2, along with ACE2 as a host cell protein, and using the protein kinase, only activates cell signaling and not any other enzymes or any other host cell target protein works like that. It's only the SARS-CoV-2 that's currently causing the pandemic that has this kind of cell signaling happening. And we also tested the HIV, Ebola, Zika, and we fortunately found one good thing, that is HIV. As I said, SARS-CoV-2 elicits cell signaling, but not the other cells. But we were surprised that HIV also elicits cell signaling by activating the STAT1 and the cytokinin expression. So we have two things now. One is a SARS-CoV-2, and the second one is a HIV that elicits cell signaling that could also cause severe effects. And we also tried to search for West Nile, dengue, influenza, herpes, and we did not find any such data, supporting our thesis. So what's happening in HIV? So we know what's happening in SARS-CoV-2. Now, what's happening in HIV? So we heard, or we know, long-term surviving HIV patients have a high incidence of the pulmonary arterial hypertension, which is also accompanied by the abnormal growth of lung vascular smooth cells and also the endothelial cells. Why is this happening? What is causing all this? Again, it's the HIV's protein. That's the outer protein that activates the GB120 when entering into the human arterial smooth cells. And that results in developing such pulmonary arterial hypertension and also abnormal growth of lung vascular cells. So what's the conclusion? We know, in addition to SARS-CoV-2 spike protein, only membrane fusion protein of SARS-CoV-1, that is the previous version of SARS-CoV that we're having now, and the HIV has been reported to elicit cell signaling in host cells. And we also know the HIV positive patients have high incidence of developing pulmonary arterial hypertension. And 10. Now, the important thing, the viral membrane fusion of proteins and arterial fragments of HIV or SARS-CoV-2 may elicit cell signaling in lung vascular cells. So in addition to just getting into the host cell, in addition to just getting into a human cell, it also triggers the pulmonary vascular remodeling, and that's why it causes severe adverse effects. And that's why we are still not able to control the lung infection of the SARS-CoV-2 virus. And as I said, we could not find any other reports of MERS-CoV or Ebola, or even though MERS-CoV is is a family of COVID, we are still not able to find any data that shows the cell signaling or the triggering of cell signaling. As I said, no data for Ebola, Zika, West Nile, dengue, herpes, or influenza virus. Now, question. So we know. So can anyone tell me, like, what are the vaccines that are coming up? What are the vaccines of COVID-19 are made of? Can anyone just unmute yourself? Anyone here? No?
Ma'am, it's a Covaxin and Covishield, ma'am.
Okay, those are the vaccines. So what are these vaccines made of? What vaccines are they? What do they consist of? That's right. It's Sputnik, it's Covishield, it's all right. What is it made of? Now it's becoming a famous word, you know. I could give you a clue. It's been a talk of the year. Okay, it's nothing but the mRNA vaccines. So these mRNA vaccines are made of. You also see my screen, right? So these mRNA vaccines are made of the spike protein. So if the spike protein itself can elicit cell signaling and it itself can cause severe lung vascular cells or cell death, how safe it is to use spike protein, how safe it is to use the mRNA vaccines on us. You know, to be honest, like, is it safe to take vaccines? What do you guys think of it? Do you think it's safe? Sorry, I can't see your chat, so it should be great if you can unmute yourself and answer the question. Okay, I'm going to go ahead and just talk about this. So since the viral membrane fusions of the proteins are often used as basis for the vaccine development, it is always critical to further understand the role of these proteins in eliciting cell signaling that may promote adverse health consequences. But you could ask me, like, flu vaccines are safe because they are also made of hemagglutinin, that is the membrane fusion protein of the virus, but they don't affect host cells. Yeah, as we saw them, these hemagglutinin membrane fusion protein of influenza virus, they do not elicit cell signaling. So flu vaccines are really safe. But since the spike protein can elicit cell signaling now, how far it is safe to take these vaccines that are developed based on the spike protein? That's still a question, and they're still discussing about it, and there are still researches going on. And this formulation is being taken by Pfizer and Moderna, and they are working on it. So I'm happy that we found something interesting that could help, in a great vaccine research study. To move on, the advancement in COVID vaccines. So this is something I'm working on right now. So there are vaccines that are developed for COVID, but mRNA vaccines take really long time for the production because you always need a stable mRNA are never stable. So we always need a stable cell line for developing a COVID-19 vaccine that's based on mRNA. So the customers are now asking us for a high yield cell line, a high quality product, or a rapid time frames, which is they obviously need an end-to-end solution for this. So we have something called as the talent technology that we are really working on. Everyone are so obsessed with the word CRISPR right now, but you would have probably not heard of the talent technology because it's not that famous as CRISPR is, but they are as great as CRISPR. I will talk about that in the later slides. So I'm currently working on this talent technology. I'm working on the site-specific integration or integration with safe harbor sites, stable expression, etc. So consider you have a human genome and it has different safe harbor sites. So instead of when we develop mRNA vaccine, since it's really big, the genome is really big, we need to target the real big genome. We need a different safe harbor sites to develop vaccines. Just with one safe harbor site, it's going to take us more than 1.5 years to develop a vaccine. So what we have concluded is we are trying to insert multiple landing pads or multiple safe harbor sites in the site-specific using site-specific integration and knock in different genes or multiple genes at the same time. So this would lead us to develop a vaccine in less than 6 months. So we could see a lot of the you really have stocks of any of the vaccines right now. We have vaccines everywhere. That's seriously because the vaccines are really unstable and the storage conditions are really bad. And that's why we thought of starting this project, and it's called as Project Pasture. That we are working on, and I think this would be implemented in the next Pfizer's um, third dose, that is the booster shot. I hope this comes out real good, and we have also claimed a patent on this. So I hope this works. And now let's talk about talent technology. Okay, what's talent technology? So what's talent actually? So it's the transcription activator, like effector nuclease, or that's why it's talent. So these are also restriction enzymes as CRISPR that can be engineered to cut specific sequences of DNA. But a CRISPR is preferred tool for the gene knockout studies and screens. However, if specific gene editing happens, repairing or inserting of a mutation, for instance, is a goal, then talents are real best for it. So we compare the cleavage efficiency of the talents and the CRISPR, and we see that the talents has real high cleavage efficiency. So what's cleavage efficiency? If cleavage efficiency is high, you could insert a lot more genomes, lot more specific sites into the genome. If your cleavage efficiency is low, you could end up losing two or three multiple landing pads when you are knocking in everything together. So that's why we chose talent for this. And why it is always like CRISPR, 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 and why not people talking about talents? So we also found that the CRISPR works better in less likely wounded regions of the genome, but the talent can access those genomes in the heterochromatin region better than the CRISPR. So CRISPR is kind of, it could just work, if you give them a good food they would eat, they would not make it better and eat. So that's why we have chose talent. So this could just dig into everything, and it can even get into the heterochromatin region and work real better than CRISPR. And as I said, we also saw that the talent has a high editing efficiency or the cleavage efficiency when compared to the CRISPR. Now, where is this COVID-19 taking us? What will be the future of our biotech industry after COVID-19? So we know, the public health experts, the healthcare professionals, government leaders, investors, everyone, or even the general public, they started looking for biotech industry. There were a time when, back in 2014, when I used to say, what are you doing? If someone asked me, what are you doing? I would say biotechnology, and people would ask, what's biotechnology? I've never heard of that word. I'm not sure if anyone is asking this question right now, but I've heard that question so many times. And I've struggled, in my first year, I would just struggle answering them. Oh, it's just biology linked with technology, and that's called biotechnology. That's the only word I know about biotechnology. So, but now everyone knows what biotech industry is, and they really respect biotechnologists right now. And this biotech industry is just a game-saving thing, or the game-saving vaccine against the novel coronavirus that's fearing our surrounding, and that causes all this COVID-19 outbreak. So this biotech industry has always played a critical role in developing and shipping COVID-19 vaccines all around the world in the record time. So without the industries, without the manufacturing departments, we would have not done this. And without, a proper manufacturing or R&D department, this would have never accomplished. So I think you would have heard about the Biotechnology Innovation Organization. So this said, biotechs are the main role in this pandemic. As I'm proud to say, we are all, the main role in shaping this industry, shaping the environment, or shaping the innovation. I would say you're all in the right place right now, because industry is just booming right now, and the R&D and all the companies are just wide open. Everyone is just trying out, even the companies, even the plant-based industries is now trying to work with viruses and bacteria. After these COVID things started, everyone is just switching to infectious diseases from the agricultural field. So this is just a booming industry, and I would say you are all in the right place, the right time, and I hope you get good number of openings in the R&D industry in the upcoming future. And now, why is this all of a sudden everyone is shifting to the focus? Why everyone is focusing on infectious disease research? Why is this happening? There are like four important things of why this is happening and what would be the future of biotech industry after the COVID ends, or how this is going to continue, how this is going to work out. So during when this COVID-19 pandemic started, like as you know, a large number of biotechnology companies shifted their focus to infectious diseases. There is more than I guess there were more than like 191 companies that were currently working on COVID research for getting a vaccine out. So more than 800 companies, 800 clinical trials of different vaccines have been passed, and different drugs have been passed in these 2 years. This is like this has never happened in the history before. So even companies with no previous experience in infectious diseases are now taking up this, this chain. So regardless of whether they are attempted by small or large scale biotech companies, eventually they should really be commended because they are taking a chance, giving it a try, and develop a vaccine. So this complexity of COVID-19 disease has allowed a role for many types of drug mechanism to be effective. So in terms of new therapeutics and vaccines, we are only beginning to see the benefits of new emphasis. We are still yet to go. So as a result of pandemic, organization has now shifted their focus completely to clinical research. So where would this go? What would be the end of this? We could see a lot of virtual trials that's coming out. We have never had any virtual clinical trials earlier, but this time, yes. So digital trials are likely to become the new rule. They would just rule everything, the virtual trials, rather than exceptions, not too distant future, giving concerns or anything, or even the clinical trial interruptions or postponements. Nothing is happening. We could just it's like real quick. You could just get a phone interview to have a clinical trial check with the FDA. It's as quick as just a company call. This was never the situation earlier. So we have really advanced in this, and even the US or the Indian Food and Drug Administration have been a real great job, are working real hard, are real quick in accepting the clinical trials. So this was not the case before. And friendly FDA. So as you know, getting a drug into the into the market is really hard. Passing the clinical trials was really, really hard. They would take years and years. By the time they release a drug for a disease, that disease would have just gone. It makes no sense to release a drug after 10 years of them causing any disease. So that's always been the case. But now, in last 1.5 years, the average time for a drug to get approved in the market if we produce proper clinical trial results are 48 hours or less than 48 hours. And the imports and export, it's even though the drugs pass the clinical trials and the FDA approves it, the export and import have always been a problem. But now, it's not at all a problem. You could send anything and everything if you have the proper papers, and that's all. No questions, nothing. So that has increased, and the supply chains. I remember if I just ordered a kit when I was in ICA, I would just order a kit or a PCR kit in Turbo Fisher Scientific, which is the company that I'm working now. It would take like more than 2 or 3 months to just send me. Or if I ordered a primer, they would take, I have seen like 4 months, they took like 4 months to just ship me a primer, my own company. But now it's like real quick. I get it like in 1 week, less than a week. So that's how it is now. So everything is just beyond our thinking. So the future of biotechnology is really strong right now. Um, we envision a day when breakthrough drugs lead to a world, without cancer, AIDS, or Alzheimer, a world where, there is sustainable development that will tackle the energy, food, or environmental needs without compromising the Earth's resource or its future. So, yeah, I that's all I have for today, and I'm open for any questions, any comments, anything you need to discuss about how the industry works, or yeah, I'm open to anything.
Uh,
Okay, I do see lots of questions. Uh, can the same vaccine can prevent multiple diseases like the same plant give rise to potato or tomato? It's like making a hybrid vaccine collaborating multiple genes. Is it possible, ma'am? Okay, that's a good question. But no, the answer is no. So there is no such viruses that has the same genome. If you just take SARS-CoV, each mutated version has different genome, and we are still not able to target all the get all the new developed mutated genes into the same vaccine. Then how is it possible to develop a vaccine that could collaborate, or you could call it as a hybrid vaccine? We could never develop that. For now, I don't think it's possible, and I don't think it's possible in future. Maybe if it's the same family, when maybe if the genomes are similar, we could target. But when there are different genomes, it's really difficult to accommodate all the genomes into the same vaccine.
uh, Participants, if you have any questions, you can post your questions in the chat box.
It can be any silly questions. It's okay. How did you really want to go to US? Why did you move to US? Any silly questions is okay for me. It's fine just opening up and asking questions.
Uh, I hope everyone is clear with your presentation. Okay, shall we conclude the session?
Yes, ma'am.
Yes. Uh, Thank you, Ms. Sri Jayalakshmi. Sorry, I wish to call you by that name. So on behalf of our management, Sri Ramakrishna College of Arts and Science and Department of Biotechnology, and on my own behalf, I would like to extend my hearty gratitude to you to accepting the invitation in your demanding schedule and spending time in your anomalous timing. I think it is almost midnight for you now. And also thank you for sharing your acquaintance towards your experience and SARS-CoV-2 vaccine production. Best wishes for your upcoming projects, and thank you so much once again.
Thank you so much. I would really love to or like to extend my thanks to the Principal and Secretary, Dr. B. L. Shivakumar, and Associate Professor and HOD, Dr. Swash Kumar, and Dr. Chima, and other faculties and staff and students who are patiently listening to me and giving me this opportunity. I'm open for any questions anytime. You could just shoot me an email, or you could connect me through LinkedIn. I'm just going to send my email in the chat box. If you have any questions, if you need any guidance, please feel free to contact me. I'm just open to any silly questions. I would usually reply to emails like in 3 days, but if it takes a bit long, just please chat with me. But I will just get back to you as early as possible. And yeah, it's been great. You all have a great platform. Please utilize them. Please open up, ask questions. I think Sheila ma'am is like, oh, என்னென்ன வேலைலாம் பார்த்தீங்க நீங்களே கேட்கல இப்போ நீ இவ்வளோலாம் வசனம் பேசுறியான்னு நினைப்பாங்க. But yeah, I could read your mind, man. Yeah, but I'm
No,
so
you're
happy.
always a good student, Ivana College. So never mind.
I'm happy to hear that. I
Yeah,
remember
ma'am.
Sheila ma'am just like yelling at me, can you just shut your mouth for at least 10 minutes in the class? I see her telling this to me like for all 3 years I've been there. So yeah, but you have all helped me shape better in my life, and thanks to SRCAS and college for everything. Thank you.
Yeah. Thank you. Thank you. We miss you too.
Yeah, I miss you too, man. Thank you.
Yes, if possible, leave your email ID in your chat box now,
Yes, I'm just
so
gonna
that
say it
someone
right
contact.
now.
Meanwhile, the participants, an announcement. The second session will be starting by 2 o'clock, and the resource person is Mrs. Dr. Lakshmi Mohan from Department of Food Technology, Saint Kitts Engineering College, Kottayam. So she will be presenting the guest lecture on the topic of future of food, the opportunities and challenges in industry, biotech industry. So hope to join everyone. Thank you so much. Jay Lakshmi, if possible, if you can spend some time, you can also join the session.
Yes, it's probably midnight for me, but I would just like that. Sure.
Okay, then probably I should say you good night now.
Yeah, thank you.
Yes, thank you so much, ma.
Have a
I'll
good one,
contact you
guys.
in the mail.
Yeah, sure. Thank you.
Yes, thank you. Thank you so much. Participants, thank you so much. You can leave the meet now and you can rejoin the same Google Meet link. The session will be starting by 2 o'clock and you can join the session by 1:45. Thank you so much.